Hello everyone, my name is Matthew Abrams and I'm the Director of Science and Training at INCF. I'd like to welcome you to our contribution for this year's Brain Awareness Week, which focuses on brain data science and how it contributes to our understanding of the brain. Brain data science, also known as neuroinformatics, is the branch of neuroscience devoted to the development of technologies that enable neuroscientists to identify, analyze, model, organize, and share their data. It sits at the crossroads between computer science and neuroscience, applying neuromorphic engineering, computational neuroscience, and ontologies to neuroscience databases to integrate, visualize, and quantify neuroscience research. Brain data science is important to neuroscience because, as you know, neuroscience is a multidisciplinary science that combines fields such as physiology, anatomy, molecular biology, developmental biology, cytology, computer science, and mathematical modeling to understand the fundamental and emergent properties of neurons and neural circuits. Neuroscientists are analyzing data from the level of the entire brain all the way down to gene expression. Brain data science produces the technologies that enable neuroscientists to integrate and analyze the data from the multiple levels of the brain produced by the subdisciplines of neuroscience. And this helps us to further understand the brain and the role of the nervous system in health and disease. So what are the challenges we're facing today? Big data. I know we've all heard of big data, but what is it exactly? Big data is larger, more complex data sets, especially from the new data sources enabled by brain data science. These data sets are so voluminous that traditional data processing software just can't manage them. But these massive volumes of data are needed for us to fully understand how the brain functions. So this is where you come in as citizen scientists. Neuroscientists need your help. How can you help? As I mentioned, brain data science is the field of neuroscience that develops the technologies that enable neuroscience to do their work. And guess what? They've developed tools to help high school students, undergraduates, and citizen scientists like yourselves contribute to neuroscience research. My colleague Bill Grisham from UCLA will give you a talk on some tools and apps and games that you can use, regardless of your experience level with neuroscience, that you can use today to contribute to neuroscience research.
To introduce myself, I am Dr. Bill Grisham. I'm affiliated with the UCLA Brain Research Institute. I am the iNeuro project lead, which is about curricula in neuroscience, and I am very proud to be the INCF Deputy Chair of their Teaching and Education Committee. So let's get on with it. I'm going to show you a few websites today that will enable you to do this. The first one is hosted by Dr. Ariel Rokem. He is a colleague of mine on the INCF Teaching and Education Committee, and he has a website that's called Fiber, and what you can do there is actually make some judgments for him. So we're going to switch over to Fiber. So here you can see the Fiber tutorial, and what you can see here is the fact that a lot of mental illness is detected between 5 and 21 years of age or so. And what he has is a huge collection of brain scans from adolescents. And the idea is to understand mental health. So what you would be judging on is a whole bunch of scans he has. And why does he need you to help him? Because there are thousands upon thousands. So you're basically the quality control people for him if you participate. So you really will be participating in science if you make these judgments for him. Now, what he's doing is he's looking at the fiber tracks or the axonal pathways in the human brain. And what the computer has done here is it has colored up the big bundles in the human brain. And what you're seeing right now is some bouncing between the, so as it's going bigger, you're going down from the top. And you can see all these beautiful fiber tracks. Now, sometimes you gotta check out and see if the computer does a good job. Okay, and so the color bundles should be continuous and mostly fill in the dark spots. And maybe let's go back up and look at a brain here. So this kind of MRI scan, the dark spots, oddly enough, are called white matter. They would be white in the brain if you saw it raw. By the way, this is a brain in somebody's head still. So all this is what we call white matter. The gray part, luckily, is gray matter. And so what we're going to want to see is they should mostly be continuous and mostly fill in all of those dark, deep, dark gray things. Just to get you oriented a little bit, this is part of the corpus callosum. So is this part. One of good quality. So if you're actually playing the game, you'd give it a pass. Okay. This is one that's going to fail. Okay, so one thing is there's some dark bands across it, which means the scanner and or the computer were going a little wonky and things weren't great that day. What you can also see is there's a lot of scattering. The color isn't continuous as we go up and down from the brain. Anyway, this is his tutorial. So he trains you how to do it. And then as you wish, you can go and actually play the game and help him sort out brains that are usable for his research. You can be a participant. So go ahead and do it. Another way to participate in citizen neuroscience is by playing a game called iWire. This is from Dr. Seung's lab at MIT. What Dr. Seung is doing is he is mapping all the neurons in the retina. Why does he need help? Because there are tens of thousands of these cells. And as you can see, this is just one of them. Look at all these elaborate dendrites all over the place and all these amazing connections. You can see the little spines where it's probably getting synaptic input. And this is just an example of one of the thousands upon thousands of cells. Show you a little bit about the game. He is going to try to train me to recognize the little pieces of this dendrite within all of this electron micrograph. Each one of these enclosed areas is a little bit of cell. What I can see, the aspects of the cell change. Okay, we're going to try clicking on something's clearly not correct. Oh, geez, look. Okay, so that's clearly another cell. So anyway, that's the game. And you can score points. You get on the leaderboard. It's like any other game except for you are participating in citizen neuroscience, citizen neuroinformatics. The next thing that I am going to share with you is the bioimage suite for web. And the exciting thing for me about this is that you can examine brain scans of real people who are walking around. These are not cartoons. And you can do it on the web. This software will work on any computer. So let's go check it out. So here we are at the bioimage suite for web. And as you can see, there's just a whole bunch of tools here that can be used. I'm going to use that particular tool that I just clicked on. And now it's coming up. It always gives me this warning. I like warnings, so I'm just going to close it right now. And what I'm going to do is I happen to have a scan tucked away, and I am going to just bring it in and drop it. And that's all you need to load up a file. So I one of the things you can do here is you can grab these crosshairs and explore the brain. Now I'm going anterior, and notice how this view changes as I go anterior towards the person's nose, or if I could go posterior, let's go look at the person's occipital cortex. Here we're seeing occipital cortex in a couple of different views, including down in here. We can also go superior, see how the brain changes. Here you can see now we have a lot of white matter with gray matter fringe on it, and check it out. This is one of the favorite things that I like to go in and target. Keep all that good. So let's just take my crosshairs down here so I can show you. This is the hippocampus, of which you may have heard. It seems to be responsible for consolidating, that is to say, changing our short-term memories into long-term memories. And just anterior to it, by the way, this is anterior, you can see the amygdala, this blob right here, is really involved in emotions and lots of bad science fiction. Okay, so one of the things I like to do is I am going to segment, and what I'm going to do is I'm going to segment the gray and white matter. So I'm going to do that right over here in this tool, and I'm going to say segment image, and it's going to say, okay, 3 classes. So it's going to be gray, white, and this black, which is the ventricles. These are actually literal holes in your brain that everybody has, and they're full of cerebrospinal fluid. Okay, yes, all right. So now you can see that the gray matter has turned this brilliant red, where the white matter is this sort of orangey yellow. Okay, so something I can do here is I can copy this image, copy that, and then I can go and I can paste it into another one of their tools, and I can paste in the brain that I segmented. Ah, there it is. Beautiful, beautiful brain. And so what I can do now is I can get some quantifiable data about this brain. So I'm going to go here. I'm going to go to VOI analysis. This tells me the average intensity, which I don't think so. Here's I believe that's the gray matter because it was a bright red, and here's the white matter, and you can get some insight as to how big they are on this particular brain, and also you can export it as a .csv file, which you can open in some kind of a spreadsheet and take data. So this is just a handy little tool. I use this at UCLA with my students, but it's available to anyone runs on any platform. So you might wonder, okay, great, but where am I going to find some scans? How can I possibly lay hands on some scans? Let's talk about that. There are scans that are readily available to you. They're out on the web for free. And one of the sites is available to you is the Open Neuro. There is a public dashboard. Public dashboard is always available to you. And I should hasten to add that Open Neuro is partners with INCF. So go to that public dashboard. You're going to find sets of scans. You can download an entire study if you want. Also, if you would like to learn some more about brains, there is the Allen Human Brain Atlas Explorer, which you can download for Mac or Windows. So you will get a device of sorts that you can use to learn about the brain. So there are ways to take a deeper dive into things, and one of the places that you can do that, of course, is by going to the INCF, or the International Neuroinformatics Coordinating Facility, and we have TrainingSpace, and there are actually full courses if you want them. again, it's a deeper dive. So, but if you want it, it's there. All kinds of neuroinformatics. I should hasten to add that everything I've talked about, including the Allen Brain Atlas, is all free to you on the web, and all of our training is free to you on the web. So go ahead and get it. I hope that you're inspired and you can do some citizen science. So thank you for listening, and go do some science.
I'd like to thank you for joining us for our contribution to this year's Brain Awareness Week. If you'd like to learn more about brain data science, please click the Learn button. There you will find a list of introductory lectures and tutorials to help you get started with the field. If you're interested in learning more about the games and the apps that were presented by Bill Grisham, please click the Play button. There you will find demos and tutorials for each one of the tools presented, as well as a few others. And what good are those tools if you don't have the data? So click the Find Data button. There you will find links to repositories where you can find open data sets to download and play on those games and apps. Again, I'd like to thank you for joining us for this year's Brain Awareness Week 2021. Thank you.